But NeMO Analytics is a place where we try to bring together as much data as we can on this and other sort of cellular contexts. And I just wanted to show you briefly a snippet of what's here. So as Alex mentioned, Noelia's data isn't public yet because they're still in revisions, but Alex's data is out there, and Alex and I worked together to get this data onto NeMO. And you'll recognize this UMAP from the publication. Here it's actually split into 3, where each of these is a time point. This is 0, 30 days, and then 60 days. This is R-spondin 3, which is one of the things that Alex mentioned in his talk, is that at early times, some of these cell lines pop up different organizers of different strengths and then influence the later cell types and lineages that emerge. And R-spondin 3 is one of several Wnt signaling modifiers that appears in the hem. And here it's actually interesting to see, get my pen out. So this is the map of R-spondin 3 in all of the samples. So this is the entire UMAP. The colors in each of these 3 other ones are only the cells from those times. So it's clear early on that there's one area that has a lot of R-spondin, and later it actually comes up in a different area. So that's what these 2 blocks in the big UMAP are. They're actually separated in time. There's also notably a little bit later up here in the UMAP. I don't know if you can all see that, it's a little small, but you can zoom in if it were on your own machine. So just to point out the kind of thing we can see here, this is separated by time. We could also separate it by autism and control. And actually, Alex and I were working together to try to make some visualizations, because the additional family structure in his data sort of also makes you want to be able to see all sorts of different things. And then there's macro and normal cephalic within the ASD cases. So in the coming week or 2, I might send another link if Alex and I can come up with some more views where we can chop up this data. But I'll send some links after to the Slack channel so you guys can explore this. And so the way this website works in general is like this. You can ask for a particular gene. You just enter it up in the corner. This is Alex's data, but I've popped up a couple other data sets here. This is an in vivo data set from Poliudakis, and that's the Geschwind lab from 2019. So you can also see here, there's actually this later population. This is in differentiated neurons. So that's kind of a surprise for at least how I was thinking about it in an organizer. But like I said, also in the organoids, there is a neuronal population that starts to express this R-spondin. So I don't know exactly what that is. I don't know if anyone on the call might. Here, just to show you what you can do with this system, these are organoids from Canton et al., which is a Barbara Trutlein publication, a very specific subset at a very specific time in this temporal progression pops up R-spondin 3. Again, this is probably related to a hem-like state, indicating that these cells are trying to make themselves dorsal and maybe even dorsal caudal. These organoids, actually, Noelia mentioned, this is from the Velasco et al. Paper. These are the 3-month organoids. Again, a center in the precursors, as well as a center of expression in the neurons later. This is from the Usquiano, the more detailed time course that Noelia also mentioned. And again here, there's a very, very specific population of outer radioglia, or at least what the cell type was called outer radioglia, that's expressing tons of R-spondin 3. And again, at the end of this trajectory where the neurons are, there's another population. I do not know what is going on with this in the middle. There seems to be a third one.
Carlo, if you open if you look at the R-spondin 3 expression in the monkey Mikhail et al. 2023, you can see that the R-spondin 3 is also expressed by chiral cells and also a little bit by posterior neurons. R-spondin 3 is expressed from the by the cortical hem at the beginning, but also other cells later in development, the neurons later in development, keep, especially from the caudal neurons, keep expressing R-spondin 3.
Thanks,
So,
Nico.
so what you are seeing here, I mean, I don't know now the annotation of your cells, but you know, in the previous data set, you can see ASP23 maybe is expressed by specific neurons, right? Higher
Yeah, and I think
occipital neurons.
yeah, I think this tiny cluster in the middle actually has some green in it over here on the side. So that you mentioned that Cajal-Rotzia.
Right, exactly, right.
Very cool, very cool. So just to bring that up, we can pull up Nicola's data quickly, I think, here. I'll bring it back down. So I'll do the same R-respondent-free. While that loads, we'll come right back to
Actually,
it.
we showed that ASP23 is one of the genes which is kept from early progenitors to
Nice.
until more mature neurons.
Yeah,
In the occipital area, I'm talking about these are regionalized.
interesting.
It's a regionalized gene, right?
But it's not through everybody though. It's interesting. It's through the trajectory at different stages, but in very discrete subpopulations. Maybe it's still serving some kind of guidance role in a different way. So here, this is still loading, but here's Mikali. This isn't the whole data. I should have loaded the bigger one. But this is actually a later neuronal state in yours. This is around 100 days.
But these neurons are occipital. I mean, you don't distinguish here
That's right.
the region, but these neurons are occipital. Let me annotate.
Interesting.
Yeah, these neurons here must be occipital. I mean, I know because if you open the data set that we put on the paper, you can see that ASP23 is also it's expressed in the occipital neurons.
Very cool. Here, I'll open one more where we'll be able to see that, and then while that's loading, I'll show another piece of data. This is okay. So one more data set over here. This is interesting, right? That means something about the regionalization of the organoids here, where we're seeing if you're saying occipital is a specific region of the cortex where R-respondent is maintained in postmitotic neurons, it'd be interesting to see what the regional fate is in these organoids. One more thing on this profile here. This is an amazing set of genome-wide transcriptome data at near microscopic level. This is individual cells from a 16.5 brain. This is Chen et al. They don't use one of the standard spatial techniques. They do use their own nanoball patterned arrays. They call it MOSTA. But in any case, here where you can see the choroid in the ventricle, actually, this is probably the hem, and Nico, you can back me up or correct me on this, but this was the most sort of posterior caudal of the front end of the brain here, where the hem is setting up a signal. So this is not only interesting for looking at single genes as we have been, but we can look at signatures, and I will in a moment. Let's go to see if we can see Nico's data. Oh, I spelled it wrong. That's why it's not coming up. R-respondent. We can look at these organizers and Nico's data in a second here. So here's the one, but the full map should be coming up here in a moment for R-respondent3. So we should be able to see not only this neuronal, but the earlier states. This map is later. This starts at 40 or 50. So as you can see, there's tons of data sets, and I have lots of these tabs open. So there's some loading time here, but it's usually worth it.
But if you if you scroll down already, so here.
Yeah, these are the
but here, and then when you show the panel of the patterns here, you see ASP23 is basically caudal. It's a posterior. So this is yeah.
Blazing. So these are all subsets of his data,
right.
Of the macaque atlas that were different subsets that were used in the paper. These are organizers. These are the radioglia, and here's that R-respondent positive cluster of hem-related cells. And these are going to all be early too, right, Nico? This is going to be.
Of course, yeah. This is E37, yeah. So very early. Yeah.
Cool.
E37, E42. You don't see the organizer after E54, which is you know, so they're very early states. That's why I was asking to Alex that, you know, if you want to, of course, like he showed, if you want to look at the check for organizer markers, you must check early, right, at T0. He showed T0.
Right.
In fact, you know, in that UMAP, T0, T30, T6, you don't see organizer after at late time points.
Yeah, this was surprising to me. There's a second sort of newly appearing R-respondent 3 positive. We could see that. I don't think they're neuronal down here. This was in your medial cortical plate. Is Alex still on? Alex, are you still on? You might have had to go, man. So one last thing, I know we're over time. One last thing I'd like to show you guys. So you can surf all of these data sets and many, many more on NeMO, gene by gene like this. But there's also a way to enter gene signatures in both very simple ways and complex ways. So in a very simple way to show, I have the same data collection we just looked at, but now instead of a single gene up here, these are the cell cycle genes that Seurat, for instance, uses to classify S and G2M. So the sort of canonical list, we're looking at somewhere in the range of 20 genes for both S phase and G2M. So now this is not colored by a single gene, but colored by the composite expression of a packet of related genes. So S phase lights up a very specific subset here that Poliudakis et al. Realized at the time also was S phase cells. They're here, which is this signal right there. So now we're looking at this S phase list of genes across the single cells here. And in Alex's data, this is kind of cool. There are two different areas where it looks like there's a cell cycle going on. These are just the broad cell types. But there's a clear S phase going on down here and a clear S phase here. We can look, while we're looking at the other data, I'll pull up a different display of this where we have the individual times on it. In the Trutline data, you can also see it. There's actually cycling cells at different points because she has the whole time course from pluripotency back here. So this is a pluripotency S phase. This is a neuroepithelial S phase. And this is the neural precursor or even radioglial S phase up here. Kind of cool that they're separated. Here are the Velasco and Usquiano organoids. Again, you can see way back in the progenitors, this S phase is present. And here in the full section of the brain from Chen et al., it's not particularly clear, but you can definitely see it's closer to the ventricle. And what's actually really cool is when we switch over to G2M, I'll show you something neat. But maybe, yes, here's Alex's data has loaded in the meantime. So for some reason, it's changed the order of time on us here. This is time 0, where the sort of large progenitor pool is cycling from S to G2M. And then at 60 and 30, there's this second population of cells that's cycling. It's quite at a different space. It'd be interesting to try to track down what that is. But anyway, the point here is that you can, once you get lots of data sets together and you bring in genes or gene lists, and these can be as complicated as a genome-wide principal components with weights for every gene. Up here in the corner, I'll show you, there's different ways we can project these gene lists. This is just a list of genes with no weights on it. But you can get quite creative with the in silico experiment you can design when you pick a bunch of data sets to look at, and then you pick a bunch of interesting dimensions to look at in them. So one last look at this data. I've actually zoomed in. Just if you click on these images, you can say just open in a new tab to make it bigger. This is that S phase image. Oops, sorry, I jumped ahead. Open a new image. So this is the S phase image. And you can see it's actually just off of the ventricle where it's heaviest. There's some purple under that. But what's really cool is if you go to the G2M picture, right on the ventricular lining is where things are going through metaphase, which is, of course, connects directly to the central dogma of cortical development, that is where radioglial cells divide. Just as an example of a very simple gene list with an incredible high resolution full transcriptome data set can start to show you these things. Just wanted to show you that. I'll send some links to the Slack channel, and we can discuss other things in this space that you can explore in the things we already have up. And as Alex and I make more views of his data, we'll make them available for people to explore. So I guess with that, I'd just like to thank everybody for coming and now for staying late. We still have a nice crowd. I think we went from about 40 people down to about 20 as the hour ended. A special thanks again to Alex and Noelia to kick us off. And in a month, we have another session, and I'll be sending out information on that. And in the meantime, we can use the Slack channel to talk about any of these issues and to try to use NeMO to connect to data that helps us understand these issues. And if you're interested in exploring it further or getting your data onto it, please do let me know. It's great just to explore your data, but it can also be used. You can set up permanent links and actually use it as a place to make your data public for, in addition to a repository for a publication. So thanks again, everyone, and hope to see you in a month. And don't be shy on Slack to ask more questions and interact.
Thank you.
Thanks, everybody.
Thank you.